what we're doing when we're talking about epidemiology is we're talking about infectious disease from a population perspective. Okay, so a lot of infectious disease studies, when we're talking about infectious disease, we're talking about individuals. We're talking about how a particular virus impacts an individual, how it causes disease in an individual, how we treat an infection in an individual, how we diagnose an infection in an individual. And that's all obviously super useful, extremely useful. But also we need to think about these issues from an epidemiological perspective. We need to think about them from a population level perspective, because there's information that we can get from a population level that we can't get from individuals, and there's strategies that we can implement at a population level that we need to be thinking from an epidemiological perspective to put those strategies into place. So we're thinking about public health interventions. When we're thinking about the different public health interventions we want to put in place, we need to be thinking about population level information, don't we? And obviously this is super topical right now because epidemiology is all over the news just because of the pandemic we're in. A lot of the strategies we're using to try to control COVID are population level strategies. We're thinking about how we can stop the infectious agent, the coronavirus, from spreading through the population. Now, obviously, a lot of strategies to stop spread at a population level are the same as those that are used at individual levels. If we treat individuals with infections or we prevent individuals from becoming infected, then we can overall stop the spread through the population. So there's going to be a huge overlap between individual medicine and population level. But we still need to kind of take a step back and think about everything we've talked about so far from an epidemiological perspective. And we started doing that before Christmas. We specifically looked at something called the compatibility filter. Now, when we're talking about epidemiology and we're talking about the kind of population level interventions we can take to control an infectious agent, we generally split them into two categories. We split them into either targeting the compatibility filter or targeting the encounter filter. And I'm just going to run through what I mean by that very quickly at the start. So fundamentally, when we're trying to reduce infection in the population, when we're trying to control an infectious disease in the population, we're trying to lower the R number. Again, we defined this R number before Christmas. I'm sure you know what it means anyway. And R number is the reproductive number of a particular infectious agent, which fundamentally means how many additional infections can we expect from each infective case. So what's the average number of additional infections that result from each single infectious individual? And that's what we define as the R number. Obviously, the higher the R number, the more rapid the spread of the infectious agent throughout the population. The lower the R number, the slower the spread, the less rapid the spread. And the key tipping point is 1. Because if the R number is above 1, that means the incidence is going to increase. That means it's spreading throughout the population and you're going to see more and more cases. If the R number is below 1, that means the infectious agent is kind of dying out within the population. We're seeing less and less infections. And that makes sense, right? Because if each infected individual on average infects more than 1 additional people, then we're going to see an increase in cases. We're going to see spread. However, if the R number is below 1, so each infected individual on average infects less than 1 additional person, then the infectious agent is trending on a downturn, isn't it? The cases are going to decline within the population. So what we want to do from an epidemiological perspective is reduce this R number and, if possible, reduce it below 1. Again, in the news, you'll see a lot of discussion about this R number. Particularly, you'll see discussion about whether or not the R number is above 1 or below 1. And that's why, because 1 is the critical point. You could ask from that information, how can an individual infect 0.8 others? Or how can an individual infectious person or infective person infect 1.2 additional people, or 1.5? How can they infect 1.5 people? this is an average, so it's an average. Obviously, no individual person is going to result in 1.5 additional people being infected. You can't infect half a person, doesn't make sense, but it's a statistical average across a population. And then what you'll obviously see, because it's a statistical average, is we're not dealing with whole numbers. So just to clarify that. So from an epidemiological perspective, what we want to do is look down at the population, look at the statistics, find out what the R number is, and then bring the R number down. And as we said, there's two ways we can do this. We can close the compatibility filter, or we can close the encounter filter. And again, we defined the compatibility filter in a lot of detail in the last lecture. We went into some really specific ways in which the compatibility filter is targeted by epidemiologists, you know, and other individuals in medical health or medical science. We looked at some of the specific examples of strategies that are used to close the compatibility filter for specific viruses. Fundamentally, the compatibility filter revolves around the idea that a particular infectious agent can only infect individuals that are compatible. So if we can make individuals incompatible, and we discuss some of the strategies that can be used to do this, we can prevent the spread of an infectious agent throughout the population, and therefore we can reduce the R number. And again, as we'll see over and over again, there's this huge overlap between individual medicine and epidemiology, because a lot of the strategies we use to make individuals incompatible are, you know, they're a part of individual medicine. They're things like vaccination, for example. You're vaccinating individuals, but from an epidemiological perspective, we're thinking about vaccinating enough individuals to lower the R number within a population. So that's why that's how it comes into epidemiology, really. Okay, so the other filter is the encounter filter, and the idea here is similar. It's about lowering the R number, it's about controlling the spread of the infectious agent, but the way it's done is different. So with the encounter filter, we're thinking about encounters between the infectious agent and individuals. And the principle here is that an infectious agent can only infect individuals that it comes into contact with. So if we have a particular virus, any particular virus, it can only establish infections in individuals that it physically comes into contact with, right? If you never come into contact with a common cold, then you're never going to get a cold, because the common cold, the viruses that cause the common cold, they haven't physically come into contact with you. They can't establish infection. It doesn't matter if you're compatible or incompatible. There's no encounter event, so there's no infection. And again, that's the principle of the encounter filter. We're taking advantage of that. We're trying to limit the encounters between an infectious agent and individuals within the population. That's what closing the encounter filter means. And this is a very, very powerful way to stop the spread of an infectious agent in a population. Generally, the compatibility filter and the encounter filter, these are both targeted in epidemiological strategies. So it's important to remember that it's not one or the other. We use a combination of techniques. Again, if we just think of COVID-19, so if you think about SARS coronavirus 2 right now, we're trying to close the compatibility filter by getting individuals vaccinated. I think it's nearly 1 million people who've been vaccinated with the first jab in the UK now, or almost 1 million. So that's an effort to close the compatibility filter, but we're also continuing lockdowns. So we're closing the encounter filter, because that's what we're doing with lockdowns, right? We're trying to make sure individuals don't come into contact with the virus. And that's the principle of the encounter filter, reducing the encounters, and hopefully that will reduce the R number as it did with similar efforts last year. So what we're going to do today is discuss this encounter filter in quite a bit of detail. We're going to discuss the different strategies that are used in the cases of different viruses to close this encounter filter. And again, what we mean by close the encounter filter is reduce the encounters, and in doing so, hopefully bring down the R number. Obviously, as we know from the earlier lectures, different viruses spread in very, very different ways. So the strategies that we use to close the encounter filter are understandably different for different viruses. And what we have here are some broad categories of the strategies that we can use to close the encounter filter. The one at the top, killing vectors, really, really important one. If you're on my medical microbiology module, we'll have talked about this a little bit, because a lot of infectious agents are spread by vectors. Today we'll be talking about viruses, obviously, but also other infectious agents. Malaria is spread, so Plasmodia, the causative agent of malaria, is spread by mosquitoes. A lot of viruses are spread by mosquitoes as well, the arboviruses, for example. And the idea behind this strategy is if we kill the vectors, then we are preventing the spread by reducing the encounters. The way these infectious agents spread from person to person is via the vector. So if we're killing the vector, we're reducing the ability of the virus to pass from person to person. So additional people are not being exposed to the virus, they're not encountering the virus because the vectors are diminished. There's fewer vectors to physically transport the virus into their biological system. Sanitation, this is also a hugely important strategy. So a number of viruses can spread, for example, through the fecal-oral route. So they'll be ingested and then they'll infect an individual, and then they can pass through the feces. Sanitation is obviously hugely important here, because if you're having contamination from feces to drinking water or food, then that presents the opportunity for encounters with the viruses, right? So one way to prevent these encounters, so again, to close the encounter filter, is through sanitation. So to make sure these encounters aren't occurring. Again, if we pull it back to COVID, we're thinking about hand washing as well. We're thinking about individual hygiene, sanitary behaviours that help prevent the spread of the virus from person to person by preventing the encounters. If you have the virus on your hands and then you're touching your eyes, you're touching your mouth, you're potentially transmitting the virus that way. Another strategy is to remove individuals from high-risk groups by encouraging them to modify their behaviour. And this is a really interesting one. So for some viruses, there are specific behaviours that can put an individual at high risk. So if an individual is engaging in these behaviours, we can say they're part of a high-risk group. And one way we can reduce encounters in these instances is by encouraging individuals from disengaging with these behaviours and therefore removing them from the high-risk group. And again, what we're doing there is hopefully preventing them from coming into contact with the virus, so preventing them from encountering the virus. That's the principle there. We're going to talk about these in a little bit more detail later. It's a really interesting group of strategies, because very often they're very, very specifically targeted to the particular virus that they're designed to prevent encounters for. And we'll see this in a little bit, that some viruses spread via very, very specific routes and via very, very specific behaviours. And you can probably guess the kind of thing I'm talking about. Things like intravenous drug usage, things like unprotected sex amongst specific demographics. Quarantines as well. So quarantines, really, it's just another term for lockdown. They're almost the same thing from this perspective. The idea is that we're taking individuals and we're separating them, right? Either separating them into groups, but we're separating them from each other so that we can reduce encounters. Generally, when we're talking about quarantines, we're thinking about quarantining infectious individuals. So individuals who either are infective, so they carry an infectious agent, or we suspect they may carry an infectious agent, then we could define that as a quarantine. What we're doing now with lockdowns is really just locking down everyone indiscriminately, but the fundamental principle is the same: keeping individuals separated, and in doing so, reducing the opportunities for encounters with the infectious agent. And I have a little diagram at the bottom here. It's similar to the diagram we looked at in the last lecture, so I'd encourage you to compare it to that one. We start off with a highly compatible population. So this is just a normal population in which the majority of people are susceptible to the infectious agent. And what we looked at in the last lecture was how we can take a highly compatible population and turn it into a highly incompatible population by the various strategies we discussed. What we're looking at here is how we can take the same highly compatible population and turn it into a de facto protected population by reducing the encounter filter. So what you'll see is the number of blue and yellow individuals is the same, the proportion is the same. The number of actual protected individuals doesn't change. The number of actual compatible individuals doesn't change. However, what we have in the bottom left and bottom right corners is we have infective cases. So we have these two red dots that represent individuals who have the infectious agent, they're infected and they're infective, so they can potentially pass the infection to other individuals. However, what these black lines signify is they're separated from the rest of the population. Okay, and so because they're separated, all the other individuals are not coming into contact with them, and therefore they're not coming into contact with the infectious agent. So all these individuals, even though they are technically compatible, we can say they are de facto protected. They have this de facto, so in effect protection, this practical protection, because even though they're biologically compatible, they're not coming into contact with the infectious agent. So they are protected, not biologically, but they're protected by the fact that they are not encountering the infectious agent. So I'd encourage you to make sure you understand this diagram. Please compare it to the one we looked at in the last lecture. It's quite important. Okay, so what I want to do now is move through the different categories I introduced in that slide, starting off with vector control. Hopefully I outlined clearly what we generally mean by vector control. It's targeting the vectors that spread the infectious agent, and again, by doing this we can reduce encounters, and what we want to do now is just apply this knowledge specifically to some viruses that infect humans. So the first human virus we want to discuss is West Nile virus, obviously a hugely important human virus that we have discussed before in some context, but what we want to do now is discuss it in the context of epidemiology and discuss it in the context of these vector targeting strategies. So it's spread by mosquitoes, and therefore it's understandable that one strategy we use to close the encounter filter is to target these mosquitoes. And the key takeaway here is this idea of integrated pest management control. Now, it's quite simple to say, okay, we target the vector, we kill the mosquitoes. It's a lot more difficult to actually do that in real life. It's a really, really complex thing to do. Usually it takes what we have defined here, an integrated approach. So here that's defined as surveillance, elimination of mosquito breeding sites, and larval and adult mosquito management using pesticides to keep mosquito populations low. If we actually think about the practical aspect of targeting these mosquitoes, it's hugely difficult. It's an enormous task, and there's, it costs a lot of money as well. There's resources involved in this. So there's different strategies that can be used. Actually eliminating the mosquitoes not really isn't necessarily the goal. Really it's about managing the levels, reducing the levels to a point where West Nile virus infections are minimal within a population. And here we're seeing it's mostly revolving around the use of pesticides. So we have the organophosphates and the pyrethroids. You've probably heard of pyrethroids. The organophosphates you might not have heard of. These are just pesticides that are used to kill the vectors, essentially. Something we'll sometimes see is that there's a shift in approaches when there's an acute situation, so an epidemic. So when there's a huge increase in cases in a population, then the vector will be targeted much more aggressively, just because it's more of an emergency situation, because the drive is there to push an enhanced and more aggressive effort to combat the vector. One thing we obviously really need to know is whether or not these strategies work. And I've picked this paper because it's really quite good evidence that it does work. What we mean by work means do we actually see reduced incidence of West Nile virus when we successfully target the vector? So when we clear the vector, when we reduce the numbers of the vector, does that result in lowered incidence? And this paper argues that it does quite effectively. And again, we need to remember that's the end point. So reducing the vector numbers is a means to an end. And again, that's why this is quite a useful paper, because it shows that the connection is there. Reduced vectors results in reduced incidence. This paper here as well kind of builds on that. You see it's a bit more of a recent paper. And it's kind of almost reflected in the content, because what we see is this paper is introducing some more modern approaches, so things like modeling. There's a tendency to think of just spraying a large area with pesticides as being quite archaic, as being quite clumsy, as being an old-fashioned way of doing things. And that's true to an extent. You'll notice the other paper was discussing ultra-low levels of pesticides. There's a reason for that, because there is a desire to use the lowest level of pesticides possible. No one's a huge fan of pumping out huge amounts of pesticides into the environment for obvious reasons. So a lot of papers will focus on the minimum you can use. This paper has actually gone a bit further, as I've said, using quite modern approaches to think about modeling. And what they've essentially done is determine some of the different factors that can be used to predict the presence of West Nile viruses within mosquito populations. So the idea here is this information can be used to design an intelligent model that will allow us to predict epidemics and switch to an aggressive approach as and when it's needed, or even in advance of when it's needed, because we're predicting when an epidemic might come, because we're predicting what environment might result in West Nile virus being present in mosquito populations, present in levels that could result in a human epidemic. These are the kind of things you want to be thinking about when you're doing your assessments. Saying that mosquitoes are targeted because they're vectors and they're targeted with insecticides is true. It's accurate, but actually getting into the nitty-gritty of it, discussing the best way to do that, discussing modern approaches like this, it's taking it to the next level, isn't it? Yellow fever as well. So yellow fever is also a very important human disease caused by yellow fever virus, and it's an arbovirus, so it's something we also need to discuss today. It's also spread by mosquitoes, so the vector is a mosquito. So again, the target for these strategies is the mosquito. So the idea is, if we're targeting the vector, we can reduce encounters of the virus with humans. And it works, as we can see from this paper, it does work. However, there's a wall that's hit. So it works to a point, however, it doesn't achieve elimination. And this is a really important point here, because we see this with some other infectious agents as well. We've actually discussed it in the past. It's similar to influenza. So you remember we defined influenza virus as being ineradicable, and it's ineradicable because even if you treated or cured every human in the world of influenza, of flu, so there's no circulating influenza amongst humans, it would jump straight back from birds like that, because the birds are the natural reservoir. So influenza virus is constantly infecting these different birds. There's two groups of birds, the corydoforms and the enceroforms, that are constantly being infected by influenza, and these influenza viruses will jump across to humans. So even if we cured influenza in all humans, it would come back like that. So we tend to consider it ineradicable. Yellow fever virus, there's a similar situation going on here, where if you reduce the number of mosquitoes to a point where you're not seeing human infections within a population, it's not enough to eradicate the virus. It's not enough to eradicate yellow fever. because there's this second life cycle that it has, which we call the sylvatic life cycle, where it goes from NHP to NHP, and NHP means non-human primates. So you can think of this as being the natural life cycle of yellow fever virus. It's being passed from non-human primate to non-human primate. When it's passed to humans, it's almost by mistake, and that's where it causes yellow fever disease. Even if we're reducing the levels of mosquitoes that pass yellow fever virus from non-human primates to humans, you're not going to eradicate the virus, because the virus is still going to survive in these non-human primates. It's still going to survive in its normal life cycle. Further complicating this is the fact that the mosquitoes that pass the virus from non-human primates to humans are very often different to the mosquitoes that pass the virus from non-human primates to non-human primates. So even if you really reduce the transmission of the virus from non-human primates to humans, by targeting the mosquitoes that mediate that process, you're not targeting the sylvatic life cycle. You'll see this referred to as the jungle life cycle sometimes. That life cycle is still going on. The virus is still being passed from non-human primates, so it still exists. It can't therefore be eliminated, or so far it's proven very difficult to achieve elimination, and it hasn't been achieved because of this fact. Here we have dengue fever virus, and principle is the same, targeting the vector, targeting the mosquitoes that act as vectors for dengue fever virus. I'm bringing this example up because it's an example of biological control. So as I inferred earlier, and I'm sure you know, using insecticides, using pesticides can be problematic from an environmental perspective. So biological approaches are often beneficial. Biological approaches have been used with some success to control the spread of dengue fever virus in some populations. There's a genus of crustacean called Mesocyclops, which is what I'm referring to here. And Mesocyclops are what we call larvivorous, so they'll eat the larvae of mosquitoes, and in doing so they're reducing the levels of mosquitoes within the population. This is seen as good from one perspective because it's a natural solution. There's the idea that it's a bit more controlled, it's a bit more measured, it's a bit more tempered than releasing huge amounts of pesticides and chemicals into an environment. And there's certainly some truth to that, there's merit to that. However, as I'm sure you also know, there can be unpredicted effects of releasing any organism into an environment. You're potentially imbalancing the environment. You're potentially having a really drastic impact on the environment. So these biological approaches can be successful. They can also be very problematic. And what's actually been seen with these Mesocyclops in the control of dengue fever virus is, so if you'll see a load of papers in the 90s and the early 2000s kind of defining the successes, a lot of them in Southeast Asian countries, so Vietnam. I think this paper is Vietnam. In Vietnam, and they do seem to work to a point. However, there are problems associated, and it was almost abandoned in the early 2000s. You can't really find many papers, and this paper here kind of summarizes it by saying the application was not always successful, and that's certainly true. That doesn't mean biological approaches aren't good. It doesn't mean they don't work. It just means that they can be quite complicated, and there's reams of examples out there in the literature of biological approaches that have been used with varying levels of success to reduce mosquito levels, so to target the vector. If you look up, there's something called entomopathogenic fungi, entomopathogenic, so entomo, insect, pathogenic disease, fungi. So these are fungi that cause disease in insects, and these can be used to target mosquitoes, really. And again, it's the same principle. It's a biological approach to reduce the levels of the vector. And again, when we're talking about the vector and we're talking about viruses, the vector is very often mosquitoes. We can also think about larvivorous fish. So when I use the term larvivorous, I mean an organism that eats larvae. Larvivorous fish will eat the mosquito larvae. They're fish that eat the mosquito larvae, and they're widely used in some circumstances to help reduce mosquito populations. So these are just some of the biological approaches that I used. There's also, as again, I'm sure you may know, more practical approaches. So we have the chemical approaches and we have the biological approaches. Practical approaches can be taken as well. So for example, when there's an outbreak of malaria in rural communities, and again, obviously malaria isn't a viral disease. I'm just using it as an example because it's spread by mosquitoes. What is often seen in rural communities where there's a sudden outbreak of malaria is there will be a local tepid water source. So there'll be a pool of water, and actually a lot of the time it's been linked to disposed tires. So there will literally be tires that have been thrown in a pile somewhere near the community, because tires are an excellent place for water to pool. And once the water pools there, you get the tepid water, and that's where mosquitoes, that's where the larvae survive. They don't survive in flowing streams. It needs to be tepid, still water. So that's often linked to an increase in mosquitoes in the local population, which understandably is results in an increase in mosquito-borne infections. So an effective way to control the infections in those circumstances is to just get rid of the tepid water. So just clear it out, clear up the local area, and again, what you're doing is reducing the encounter filter. Following on from this, and still thinking about yellow fever, which we talked about a few slides ago, there's a really interesting example, which is the Panama Canal. So the Panama Canal is, if you think about the Americas, you have North America and you have South America, and you have Central America in the middle, right? The shipping route from one side to the other used to involve going either right round the bottom of South America or kind of over the top of Canada. There's a way that ships can weave their way through, I think. And obviously that's a huge distance to be taken. So that it was determined there would be a huge economic benefit to building a canal in Panama. So just building a canal where boats can go from one side of the Americas to the other side of the Americas and avoid making the huge trip, usually around South America. Attempts were made to do this. First off, the French tried to do this, but they didn't manage it. And one of the reasons they didn't manage it was because of the huge amount of disease there, and the disease that was primarily impacting the workers was yellow fever. If you think about the climate, we're talking about Central America. Yellow fever was spreading like wildfire, because that's where the vectors were able to thrive, and the workers were just coming down with yellow fever left, right and centre, also malaria as well. And we have a quote here from 1905 that's, it's kind of summarising the impact of these diseases on the workforce. Next, we want to think about quarantines a little bit. Again, I'm sure we all know what quarantines are at this point. What I really want you to do, though, is think about quarantines in quite a bit of depth. Now, it's one thing to say on the surface what a quarantine is separating individuals to prevent encounters, because that's what it is, right? That's absolutely true. Again, as we said, when we're talking about quarantines, particularly we're talking about taking individuals that are potentially infected or are infected and separating them from other individuals to prevent encounters, so to prevent spread. We're also familiar with lockdowns, which is a more general a kind of a level of quarantine for everyone to keep everybody separated and to reduce encounters that way. But hopefully it's intrinsically clear that quarantines and lockdowns are to reduce encounter events. Again, what we want to be doing now at level 6 is thinking in more detail and thinking about how we can evaluate quarantines. Now, we can evaluate them from a couple of angles. One is just thinking about efficacy and effectiveness in terms of, do they do what they're supposed to do? Do they reduce encounter events, and do they reduce incidence of infectious disease? The answer to this is yes. You'll see reams of literature supporting this. Quarantines work. However, there's another way we can evaluate that and actually think about quarantines and lockdowns in more detail. Think in more detail about the impact they have. There's other things to consider when we're measuring the costs and the benefits of quarantines and lockdowns. This paper is really, really good because it's talking about anxiety. It's talking about other aspects of health that are impacted by quarantines. So a quarantine doesn't happen kind of in a bubble in terms of when you're separating an individual from the rest of the population, you are reducing the potential encounters if that individual is potentially infective. However, you're doing other things as well. You're impacting them in other ways. You're restricting their ability to socialize. You're restricting their ability to gain exercise. You're restricting their ability to work. So we need to think about all these things when we're thinking about quarantines. We need to think about economic factors. We need to think about how, the population as a whole is being impacted, how the economy is being impacted. If you are going to be discussing quarantines or lockdowns in any of your assessments, this is the level of detail you want to be going into. Again, it's one thing to say whether or not they work, and even if you do that in an intelligent way and you're supporting it with the literature, and you're really showing an in-depth understanding of whether or not they work and how they work, it's another thing to have a more rounded understanding of quarantines as well. So actually understand all the other ways that quarantines can impact an individual and impact a population. This is a really interesting paper because that's exactly what it does. When we're talking about medicine and public health, and medical science in the modern world, we really do take a much more rounded approach to things. Maybe 100 years ago, it would be a case of it ultimately comes down to reducing infections. We're epidemiologists or we're microbiologists, and we want to reduce infections, and everything else is secondary. We want to reduce infections, that's what we're going to do. But now we understand that it doesn't work like that. We're not operating in a bubble in which the only thing that matters is the particular infectious agent we care about. We also need to understand the other factors at the very least, right? We need to understand the impact of these public health interventions. And that's not to say we're arguing that the interventions aren't justified. It just means we're showing an intelligent and reasonable awareness of the arguments on both sides. We're understanding the negative impact of interventions as well as the positive impacts as well. This can be quite a tricky thing to do when we understand that in many instances, quarantines and lockdowns are necessary, such as now COVID, and it can seem almost counterintuitive to argue against that. But we have to do it. It's the intelligent thing to do. It's how we evaluate effectively. We saw the same thing with Ebola in the West African Ebola outbreak. There were these huge lockdowns they didn't really call them lockdowns then. There was quarantines in West African countries where individuals couldn't fly in and fly out. There was it was a lot of things in the news about individuals flying back from West Africa that could have been infected, and was this a responsible thing to do? Was it ethical to put them into quarantine? And there's room for discussion there. There's a lot of evaluation. There's a lot of things to say. There's a lot of intelligent discussion to be had there. And I'd encourage you, if it's appropriate and if this is something you're exploring in your submissions, to do that, to have that discussion, to explore these ideas. This is just a little bit of history on quarantines. So hopefully we've discussed at some point, or you're aware of the idea of germ theory being born sometime in the 1800s, about the 1850s. You've probably heard the story of John Snow determining that the cholera epidemic in London was being caused by contamination of the drinking water with feces, and it was Vibrio cholera, the causative agent of cholera, that was causing the cholera, and that's what was being introduced into the water supply through the feces. And that was really a really significant step in understanding that infectious disease is caused by discrete infectious agents. For cholera, it's bacteria, but now, of course, we know viruses cause disease as well in much the same way. However, if we're going back, centuries, we're going back here to the plague in the 1300s. Even then, it was understood that there was benefit in quarantine. It was understood that quarantines could reduce the spread of disease. They didn't have a particularly good idea of what disease was, what it was caused by. Before germ theory, there's this focus on something called the miasma theory, which was diseases were primarily spread by bad smells. So the science wasn't really there. However, the principle of quarantine was established. The idea that, and this is a really good early example of quarantines, it was ships coming into Venice, I think it was the city-state then, coming into Venice. They would be quarantined for 40 days, and that's where the word quarantine originally comes from. Actually, the root is for 40, all right, something like that. So 40 days, and then the word turns into quarantine over time. And the principle is there. Quarantines are put into place. So quarantines have been used for a long time before the science was really understood, and they do work. Oh, and yeah, this is what we have, 40 days here. And yeah, again, just elaborating on that information, this physical separation between healthy and infected persons, and even quarantine of the ships themselves and the cargo. So again, physical separation to reduce the encounter events. This was occurring even before there was any real substantial understanding of the epidemiology of infectious disease. There was still an understanding that these practical things worked for some reason. Okay, and then if you want to think about still history, but a bit more recent, we're thinking about New York City in 1738. So kind of the early-ish days of America. Individuals, when they were emigrating to America, would go through New York City very often, and there was hesitancy about that because a lot of these individuals were potentially introducing disease into the population, and there were quarantine centres there. So again, the principle playing out again, individuals being quarantined, being physically separated to reduce the likelihood of them introducing the infectious agent into the population. And here, just a little bit more elaboration of that fact. Here we have the idea of cohorting being introduced. So we can see quarantines getting a little bit more intelligent. They're being implemented in a bit more of a targeted way, a bit more of a useful way, rather than quarantining all individuals together, like just shoving them together. We begin to see this idea of cohorting, where individuals suffering from different infectious diseases are quarantined separately, so we can almost see the understanding of the science slowly advancing. And I'm not sure I can discuss quarantines without at least alluding to the COVID figures in the UK. So what we have here is just a graph of the cases in the UK for COVID, and what we see is dips when lockdowns are put into place. And I know this is a crude way of doing it, just looking at the cases and then looking at the kind of mapping it to lockdowns, but there's a lot of literature out now showing that the lockdowns actually do work. When it starts rising, that's when we start going towards another lockdown. As we are now, obviously we've got a huge number of cases now, and that's why we're going back into a lockdown. They work, they do work. Yep, that's all I really want to say there. And the final group of strategies we want to discuss is the idea of behavioral modification. As we said, the main idea of behavioral modification is if we can encourage, and I use that word carefully, encourage, because there's a line where we can't necessarily tell people or really force people what to do, because there's a huge ethical cost to that. What we want to do is encourage individuals to change their behavior if they are normally engaging in high risk behaviors. So we want to encourage individuals to move away from high risk behaviors, or alternatively to engage in preventative behaviors or protective behaviors. And this can be a very effective strategy. If you're in my medical microbiology module, we talked about this when we were discussing ethics. There's a lot of ethical issues here, because how far can you go to get someone to change their behavior? Where is the line between encouragement and coercion? And we'll talk about this a little bit now. This example here is to do with condom use, and the point I'm making here is if you can encourage individuals to engage in protective behaviors, so condom use, or we can flip that and say if we can encourage them to disengage from high risk behaviors, which is unprotected sex with individuals they're not in a monogamous relationship with, should we say, then there is an impact on the spread of infectious disease. That's absolutely true, and again this paper demonstrates that excellently. If individuals are wearing condoms, there is reduced incidence of a whole range of sexually transmitted infectious diseases. So the principle behind this strategy absolutely works. The main problem with this strategy is, one, the ethical issues that I just alluded to. Two, it's actually quite difficult sometimes to see a change in individuals' behaviour. So individuals are often resistant to changing their behaviour because people they like doing what they want, right? That's why they do it. So if you want individuals to change their behaviour, you have to you have to motivate them or educate them or encourage them. You have to do something. You're trying to instigate a change, and that can be very, very difficult to do. Definitely not impossible, but difficult. Add to this the fact that many high-risk behaviors are complex behaviors. So intravenous drug usage, very, very complex behavior. Drug usage alongside unprotected sex. We'll talk about something called chemsex in a few slides. Complex behavior wrapped up in a lot of other things. Very, very difficult to change some of these behaviors. So even though behavior modification can be a really effective strategy, actually instigating the change can be very, very difficult, but again, not impossible. So what we have here is quite an interesting paper, which is going into some detail about what I've just said regarding condom use and how it's not as straightforward as it can seem. So on the surface, we can say wearing condoms is a great preventative measure. It's a safe behavior to engage in. If we can encourage individuals to, wear condoms, at least in sexual encounters with individuals they're not in a monogamous relationship with, we can reduce the incidence of sexually transmitted infectious disease, and that's absolutely fine. That's a valid thing to say. However, there's layers of complication behind that. There's layers of reasons why individuals might not engage in condom use, and some of those are defined in this paper here. This paper is not just referring to condom use in non-monogamous sexual encounters, it's also monogamous sexual encounters as well, but the principle is the same. So some of these reasons are the respondent claimed they knew a partner's sexual history, because another form of birth control is being used, and the belief that condoms limit sexual pleasure and enjoyment, and trust in the partner. So they trust the individual, so they don't see themselves as participating in a high-risk activity. Other reasons include inaccessibility of condoms, partner disliking condoms, or personal condom dislike. So they just don't want to wear condoms, or sorry, they don't want to engage in sex with condoms, or their partner doesn't want to engage in sex with condoms. And so what we're seeing here is, if we peel back the layers of what looks like a simple solution, we're seeing it's actually very, very complicated, because there's all these barriers to condom use that can exist. And these are things we need to think about. These are things we need to evaluate. And if we're looking to submit a really strong assessment in this area discussing these things, these are things we really need to, pull apart and understand and potentially offer solutions to. It's not enough to say we need to encourage individuals to engage in condom use to reduce the spread of sexually transmitted infections. That's a fine statement, it's a true statement, but it's not a level 6 statement. What are we going to do about these specific issues that have been identified in the literature? How are we going to overcome these barriers? It's really important that we think about things in this level of depth. Onto the next complex behavior that we want to talk about, which I alluded to earlier. It's a behavior called chemsex, and it's well defined in the literature. It's a behavior which involves engaging in drug use and then engaging in sex alongside the drug use. So a lot of the drugs will purportedly enhance, various elements of the sexual encounter. So you can read the literature about this, but the idea is that individuals will engage in drug use and engage in sexual encounters alongside the drug use. Now, there's a lot of problems here. Again, and I'm talking from a scientific perspective, from the perspective of infectious disease. By problem, I mean there's risk factors here that can result in transmission of infectious disease. So drug usage and alcohol usage are correlated with unsafe sex. They're correlated with decreased condom use. They're correlated with increased transmission of sexually transmitted infections. That's just true. That's in the literature. So straight away, this is an issue from that regard. And there's also other issues as well. The chemsex is a behaviour that, if you look in the literature, is often tied up in the MSM community, so men who have sex with men. So we're talking about a demographic that is there's a high risk of HIV spread anyway, regardless of drug usage, because when we talk about men having sex with men, we're often talking about anal sex, which is a very high risk transmission event for HIV. So if we add the added complication of drug usage, we're often looking at an even higher risk of transmission. If we look at this in even more detail, we can see physical reasons why increased transmission could occur. So when we're talking about drug usage and alcohol usage, we're often talking about lowered inhibitions and increased willingness to participate in high-risk activities. So increased willingness to have sex without a condom. So a disinclination to engage in condom use often. However, we can also think about physical reasons, or physiological reasons, biological reasons, however we want to word it, why chemsex can result in increased HIV transmission. It can result in a sexual encounter going on for a protracted length of time, so for a long period of time, which can result in abrasions. It can result in micro tears in the rectum. These things are providing potential opportunities for viruses to pass from individual to individual. Again, we've talked about this in the past, how the epithelium provides a barrier for infectious agents. So the infectious agent has to pass through the epithelium, and what this paper is arguing, and other papers as well, is that chemsex can potentially result in increased opportunities for infectious agents to pass through the epithelium. And here is another relevant paper you may find interesting, which discusses the idea that condomless chemsex is associated with HIV acquisition, again in MSM, so in men who have sex with men. And this is done in a HIV testing centre in Belgium. And the key sentence here, they found an increase in the reporting of condomless sex associated with the reduce of the use of various drugs, and the reporting of condomless chemsex, so sex with these whilst using these drugs, was associated with HIV infection. So we're seeing these clear associations. So what's the solution? What's the solution? So one solution is so we're talking about a perfect world now. A perfect world, we encourage these individuals not to engage in chemsex, so we encourage these individuals to move away from this high-risk behavior, actually instigating that change in real life. So actually fulfilling that goal is difficult. There's barriers to there, just like there are barriers to inducing the use of condoms that we looked at a few slides ago. So I really want you to think about what those barriers might be, particularly if you're submitting a submission discussing these kind of issues. What we have here is another very, very complex behavior. So there's a lot of debate around this for a while about whether or not it was a real behavior. This idea, and it's called colloquially like bug chasing and gift giving. The idea is that some individuals will engage in behaviors that will they'll willingly engage in behaviors that put them at risk of HIV. Bug chasing is the idea that they are trying to get infected with HIV, or if not trying to get infected with HIV, they are willingly putting themselves into positions where they might get infected with HIV. Such a complex issue, and I'm not going to go into it now. I'm not going to, it's not something that we can cover in a single lecture, really. There's so many things to talk about here. All I really want to do is make you aware that these really, really complex behaviors exist, and what is a potential solution to this? It's so again, in an ideal world, the solution is to encourage individuals to change their behaviors. But again, complex behaviors, actually instigating a behavioral change away from this behavior, as I'm sure you can imagine, is potentially very, very difficult. This is really a fringe behavior here, so it's quite an extreme behavior, but it is documented in the literature. It does exist, so it is worthy of our attention. Okay, moving on now, we can think about hepatitis C virus, so moving away from HIV. When we're thinking about hepatitis C virus, HCV, we're thinking about intravenous drug usage, which is a very, very high risk behavior for the transmission of hepatitis C virus. Intravenous drug usage, obviously, again, another very, very complex behavior, a behavior that can be very, very difficult to change, to encourage people to change. Why? Because we're thinking about addiction, aren't we? Intravenous drug users are often injecting drugs they're addicted to. There's overlapping issues with that. There's issues of poverty, there's issues of homelessness, there's issues of mental health. Again, it's, as I'm sure you know, it's just not as simple as explaining to intravenous drug users that they should stop injecting drugs, and then they understand that, and then they suddenly stop injecting drugs. It just doesn't work like that. However, that's not to say there's nothing that can be done. It's just there's it's not a simple thing. There's not a simple solution. What we have outlined in this paper are some really, really interesting strategies that, when used in combination, have had success in reducing hepatitis C virus transmission. And what you'll see is a number of these strategies, rather than focusing on changing individuals' behavior, so encouraging them to move away from a high-risk behavior. They involve encouraging individuals to engage in uh, encouraging behavior that mitigates the damage. Okay, so that reduces the harm. So, okay, it's difficult to encourage individuals that take intravenous drugs to stop. Can we encourage individuals that take intravenous drugs to engage in needle banks, so to use clean needles, because they're still engaging in the high-risk behavior, but the damage is mitigated, the damage is reduced, because they're not using dirty needles, they're not sharing needles anymore. So they're still engaging in the high-risk behavior, but it's slightly reduced risk. The risk is reduced because we've encouraged a change in behavior, and it's not a complete change in behavior. They're not disengaging from the high-risk behavior, but their behavior has changed enough to reduce the risk. We talked at some point about prion diseases, even if it was just as a curiosity. I'm fairly confident we've talked about prion diseases at some point. Prion diseases aren't viral diseases. Prions aren't viruses. However, they're similar in the respect that they're an infectious agent, and they're passing from person to person. Kuru is a prion disease. It's caused by a prion. It's spread by it's found amongst a tribe in Papua New Guinea, and it's spread by funerary cannibalism. So they eat the brains of dead relatives, and if the relative was infected with Kuru, the individuals who ate their brain will then acquire the infectious disease. This is an infectious agent that is spread by a very, very specific transmission route. Obviously, a very different transmission route to HCV, obviously a very different transmission route to HIV, but a specific transmission route, none the same. And Kuru was essentially eliminated. I think now it has been fully eliminated from this tribe by encouraging the change of behavior. So Christian missionaries in Papua New Guinea encouraged this specific tribe, the Fore tribe, to disengage from this practice of funerary cannibalism. And in doing so, they again eliminated Kuru. So they were able to induce a behavior change in the population, and they essentially eliminated the high-risk behavior, which eliminated the infectious disease. Okay, so those are the different strategies that we can use to close the encounter filter. They're not, it's not an exhaustive list. It's just the different types of strategies we can use, and then some useful examples of those different types of strategies. Again, not an exhaustive list, but some really useful examples, and I hope you found them useful. I hope it's kind of opened I hope it's made you aware of some of the issues around these strategies as well, and it's given you the tools that you need to discuss these things in an in-depth way and really look at these problems, not just in a kind of superficial way, but in a really kind of critical, analytical way. Okay, so what I want to do finally in the lecture is discuss epidemiology and ethics. So I've touched on the fact that ethics is really, really important in epidemiology, and what I want to do now is just spend a little bit of time explaining why, spend a little bit of time discussing some of the specific ethical concerns that we have in epidemiology. So in any area of life sciences, really, ethics is inherently important, but with epidemiology, there's specific ethical concerns we want to consider. So again, we're dealing with population, dealing with population level information, and we're making population level decisions potentially. So again, there's specific ethical concerns around this. Now, the principles of these, the fundamental principles of ethics remain exactly the same as they normally would in life science or medical science. We're thinking about autonomy, we're thinking about justice, we're thinking about beneficence, and we're thinking about non-maleficence. So autonomy, we're needing to respect the right of individuals to make their own decisions and to have their own autonomy. Justice, we're thinking about the right of individuals to be treated fairly, to be treated equally. Beneficence, we're thinking about our responsibility to help where possible. So to yeah, do good essentially. So an obligation to do good. And non-maleficence, we're thinking about our obligation to not do bad. So to not cause harm. And these are the fundamental principles of ethics. What we're going to do now again is apply these to epidemiology. Now, there's different ways we can approach ethics when we're thinking about, life sciences as a whole, when we think about medical science, because ethics isn't necessarily an inherently objective thing. It's not an empirical thing. There's not always black or white, yes or no, as to when is something ethical and when is something not ethical. And if you look into the literature of this, there's a lot of debate around Kantian ethics and utilitarian ethics, which is just two different approaches to the fundamental question of what is ethical. So if we're thinking about Kantian ethics, we're thinking about the idea that things we do are inherently right or wrong. So there's a moral way to treat people and there's an immoral way to treat people, right? And that's what it comes down to. And there's no justification to behave immorally to individuals, to breach ethics. And that sounds absolutely fine, and it is absolutely fine. But there's another view or another perspective, which is the utilitarian perspective, which is the idea that we need to think about the greater good. Okay, so the immediate ethical cost can be counteracted in the greater good. So these two principles or these two approaches aren't always opposed. They're often very consistent, but they are, when we look at the details of them, fundamentally different perspectives. They're fundamentally different outlooks, even if the outcome is sometimes the same. Again, we're not really thinking about the philosophy here. We just need to be aware of those two broad perspectives, because those are the two broad perspectives that feed in to the ethics of medical science, and particularly the ethics of epidemiology here. So it's kind of the balance between what is the immediate right decision, what is the immediate ethical thing to do, and what is the eventual outcome of our decision. What are the eventual consequences? Are we doing good down the line, or are we doing harm down the line? Again, obviously, it's not the most objective thing, is it? So what we tend to do is stick to the principles which we outlined on the right. So these four principles: autonomy, justice, beneficence, and non-maleficence. These are more tangible. These are more objective in a way. So we tend to think of ethics as being principle based in life sciences. So we have these four principles, and these are the four principles that we stick to, because when we're sticking to these principles, we can be more objective. And we can fundamentally say, is the approach we're using, is the strategy we're using, breaching any of these four principles? And it shouldn't be. If we're behaving ethically, if the research is ethical, if the approach is ethical, it shouldn't be breaching any of these four principles. Again, autonomy, justice, beneficence, non-maleficence. So here we have some information on public health measures, and public health measures, it's a general term to refer to any broad sweeping measure that is implemented, usually by a governmental level, to impact the whole population. So lockdowns, quarantines, and again, with quarantines, we're not always impacting the whole population, but we're potentially impacting all individuals in the population who could be infected. So the principle here, it's these broad sweeping interventions that are what we call top down. So they're coming down from the government or other bodies like that. There's ethical issues here, because there's ethical issues around consent. You're essentially imposing interventions on individuals within the population. There's justification to do it. In the case of many infectious diseases, However, it is different dependent upon the infectious disease. So it's easier to argue that forced quarantines are ethically justifiable for diseases like Ebola, SARS, COVID, compared to influenza, compared to the common cold, compared to human papillomavirus, right? So these population level interventions, these public health interventions, for them to be accepted by the public, for them to be deemed ethical, there has to be justification for them. If we think about the lockdowns we're going through at the minute in the country and around the world as part of the global pandemic, those are seen generally as an appropriate response to the danger, to the threat, to the pathogen, to SARS coronavirus 2 and the disease COVID-19. So they're generally seen as justified, and people are generally going along with them and accepting them. If we can imagine a different situation where these lockdowns are brought in response to the normal influenza epidemic we see each year, that's just not going to fly, is it? People aren't going to accept that because it's not justified. It doesn't make sense. It's not going to be seen as ethical. So the only point I'm making here is that these public health measures, it's not always black and white whether they're justified or not. Whether they are justified or not is determined by how high individuals perceive the risk of a particular threat to be. If they perceive the risk of a particular infectious disease to be high, Ebola, coronavirus, then they're more likely to be accepting of lockdowns and these other public health measures. They're more likely to see them as justified because it's easier for public health professionals to argue that they are ethically justified, because there is a fundamental argument there that these interventions are justified because the seriousness of the pathogen inherently justifies the intervention. There's other aspects of epidemiology as well that we want to think about from an ethical perspective. Screening is one particular issue that comes up in epidemiology. So screening is slightly different from testing and treating and these other strategies that we use, because screening is a more widespread approach that aims to identify cases that would otherwise be hidden. So screening doesn't necessarily require an individual to have any symptoms. It doesn't require an individual to have any suspicion of infection or to be at high risk. Screening is fundamentally for everyone who could potentially be at risk. That's what differentiates screening from diagnosis, really. When we're talking about diagnosis, we tend to be thinking about diagnosing active infections. So there's often a suspicion of infection and then that's diagnosed. Screening, however, is a wide-scale screen of the healthy population to identify any cases that are there. There's ethical issues here that we need to think about, and one of them comes up over and over again in epidemiology, which is funding and resources. So who's going to pay for this? Who's going to pay for these things? We're talking about these broad, widespread public health interventions and population level interventions. This is really wide scale. It costs a lot of money. Who's going to pay for this? That's an ongoing question. It can depend who should foot the bill for this, really. And it's an open question. There's no answer there. Obviously, in the UK, we're fortunate to have the NHS, so a lot of this is funded through the NHS. Not many countries have something like the NHS, so they're in a slightly different situation there. So how is it funded in those countries? These are all questions we can think about. That's one of the fundamental ethical issues. The other is related to big data and information. So this screening is being done at a population level often, and there's huge amounts of data being collected. What do we actually do with this data? Who gets access to it? How is it processed? Is it anonymized? What if there are attempts to sell on this data and things like that? These are all, real potential issues that we need to think about. So again, this isn't an exhaustive list of the ethical issues. It's just some of the things we can think about when we're evaluating epidemiological strategies. There's an additional issue which is a bit unique to screening, which is because you are targeting healthy individuals, that can present another barrier to participation. So individuals may be less inclined to engage with these screening programs because they feel fine, because they're not ill, they don't have symptoms. If they're not ill and they don't have symptoms, why would they want to go through what could be a complex or uncomfortable screening process, right? One of the ways we get around this is through education. We inform individuals that they could potentially be at high risk, and then hopefully they engage in screening processes. Now, what I'm sure you're noticing here is everything I'm saying also applies for non-communicable diseases. There's a lot of cancer screening programs. And that's absolutely fine. Really, the topic we're focusing on is the epidemiology of infectious disease because we're on the virology module. However, a lot of the principles within the epidemiology of infectious disease exist throughout epidemiology as a whole, so they're consistent with non-communicable disease as well. So just be aware of that. And just to elaborate on some of these ethical issues of screening, there's the issue of consent and getting individuals to agree to be part of screening programs as well, which we've said, but it gets a little bit more complicated when we're thinking about infants, right? So we're thinking about infants who could potentially be at high risk of a disease. What are we gonna do about it? We really want to screen these infants, but how do we get consent? What if the parent doesn't consent? At what stage can it be made mandatory where the parent doesn't have the right to refuse consent anymore because it's in the interest of the infant? Again, complex issue, not really black and white, just something we need to be made aware of. This issue of consent also follows into this next element I want to talk about, which is essentially the encouragement of positive health or the promotion of positive health behaviors. Now, what epidemiologists want to do is to encourage individuals to engage in positive behaviors. So they want to encourage individuals to engage in behaviors that have a positive impact on their health, or at the very least don't have a negative impact of their health. And this is, we did talk about this earlier when we were talking about behavioral modifications, right? And what we want to think about now is a little bit more about the ethics of this. It's difficult because there's a fine line between encouraging positive behavior and coercing individuals into positive behavior. Now, the idea of coercion is the idea that you're not just encouraging individuals to behave in a positive way, you're actually manipulating them or using some degree of force to make them behave in a positive way or to influence their behavior. And there's ethical issues there, because even though you could argue that, you know, the end result is positive because they're behaving in a positive way, there's still these ethical issues there because individuals have autonomy. We know the principle of autonomy, so individuals have the right to make their own decisions about their health and their well-being and their behavior. And if they are being coerced into a particular behavior, there's an ethical cost there. It's not ideal, and it's something that's generally avoided. So this line between encouragement and coercion is actually quite important. It's also important from a practical perspective, even aside from the ethics, because if individuals feel like they're being coerced, if individuals feel like they're being manipulated, if individuals feel like their autonomy is being breached, they will lose faith in the healthcare system very, very rapidly, and long term this can result in them becoming disinclined to engage with healthcare. So these long-term effects can actually be net negative anyway. So it is really a problem if we have this coercion going on. What we have on the right here is a news story that was you might have seen it on the news. That came out recently from Spain, and it's the idea that individuals who are not willing to be vaccinated against SARS coronavirus 2 will be placed on a register, so their names will be listed. This is it makes me pretty uncomfortable. It should make most people uncomfortable, really. This is really walking on that line between the encouragement of positive behaviours and coercion. This is really right on the line. It's a really tricky one, because you can understand from an epidemiological perspective what they're trying to do. They're trying to encourage individuals to be vaccinated because there's almost this underlying thread of a threat here. It's almost a threat where if you're not being vaccinated, you're put on this list, and that's how a lot of people are going to interpret this. They're going to interpret it as a threat. They're going to interpret it as coercion. Obviously the reason why this is being done is to encourage people to vaccinate, and also to have this information on individuals who are not vaccinated. And there's perfectly valid epidemiological scientific arguments why this should be done, why it is justified to do this. But again, a lot of people are not happy about it, and the reason is what we just talked about. It feels like coercion. It feels like a breach of autonomy. It feels like manipulation. Therefore, it is problematic, and that's all I'll say about that. Okay, finally, I think I used a phrase called top down earlier. I just want to elaborate on that phrase and distinguish top down from bottom up approaches. Now, there's an issue within epidemiology which is related to the fact that inherently a lot of the decisions being made are being made from the top. They're being made by individuals who are experts in the field, who have very well-paying jobs, who are quite distant from the working class communities that the decisions will eventually impact. And there's ethical issues surrounding that. What we essentially have is a number of people who are very educated, making decisions that will impact a huge number of people who are in a wide range of socioeconomic situations. So we kind of think of that as being top down. So these few individuals on the top are making these decisions that impact all the individuals on the bottom. Again, obviously there's ethical implications around that. Individuals who are making these decisions need to be very, very aware of the specific circumstances that could potentially be impacting the real world environment in which their decisions are impacting, right? There's a tendency with epidemiology to think of people sometimes as numbers or statistics, but that's not the real world. That's just not how it works. That's not an ethical approach. So we need to take into account things like economic factors, religious factors, cultural factors, all these individual factors that will impact the way in which large-scale interventions actually play out in communities. And this is why there's this flip towards more of a bottom-up approach, which is getting individuals and communities actively engaged in the decision-making process, making these public health decisions in a way that is very mindful of the circumstances of individuals in these communities. It's just a more ethical way of doing it than having a small number of people who are, again, very distant from a lot of these communities, making decisions that will impact these communities in a very, very significant way. Okay, that's everything I wanted to say about epidemiology today. I hope we've covered the encounter filter really effectively. I think we have. I think we looked through some really good, strong examples of the ways we can target the encounter filter, and I also think I gave you the opportunity to explore it combined with the tutorial that we'll do next week. I think you've had the opportunity to learn how to really discuss those strategies in a good level of depth and in a really critical way. These papers I'm putting on the slides, by the way, I really want you to go and read these papers. There's a reason why I'm putting the references in. It's because I want you to go find these papers. Again, what I'm doing is I'm kind of directing you towards the literature. What we're covering in the lecture is kind of it's a starting point for you to go into the literature and read things in more detail. So I really would encourage you to do that. And I hope the ethics has been useful as well. Again, just giving you the tools you need to discuss whatever strategies you decide to discuss in your case study, for example. Ethics is a huge part of epidemiology. So if you are discussing epidemiology and epidemiological interventions, it will be really useful for you to be discussing ethics as well. Okay, thanks very much, everyone. Take care.